welcome. I'm Donal O'Shea. I'm a specialist in obesity in St. Vincent's and St. Columkills, and I am thrilled to be hosting this evening. I don't think I've hosted anything actually before, but I'm hosting this. Sir Steve O'Rahilly is a graduate of UCD like myself. He is currently Professor of Medicine in Cambridge and is, I mean, you know, without doubt, and he's embarrassed by all of this, but I mean, you know, he would just prefer to get up, give his talk, and I'd probably let him do that. But he is undoubtedly one of Ireland's leading scientists and greatest ambassadors, both within Europe and the US, for what we can produce from, you know, our people. So, you know, from Finglas through UCD to America, to Cambridge and to knighthood is an extraordinary story and journey, and we'll maybe get to a little bit of a chat about that after. But first of all, I'd like to introduce him and thank him for giving of his time to this final event. And Steve is going to talk about the genetics of obesity. Thank you very much.
Thanks, Don. I think I'll stand over here so I can use the beeper a bit easier. Excuse the monkey suit. I was going to say that UCD people always feel a bit daunted by Trinity, so I thought I should dress up. But I'm actually going. It's very posh here, isn't it? But I am actually going straight on to a dinner, so I didn't have time to change afterwards. So I'm going to talk for about 30 minutes on obesity, which is a topic that both professionally and personally I wandered into entirely by accident. Now it's rather it's turned out to be a fascinating journey in that, unlike most other diseases which people find boring and turgid, everyone's got a deep set of personally held beliefs about obesity, about their own body weight, their spouse's body weight, their dog's weight, and it's very hard to shake people's views because they're very firmly held and they're deeply entrenched in personal experience. So perhaps I hope I'll be able to shake some of you out, perhaps out of some of your preconceptions tonight, because obesity covers everything from a world important public health problem right down to the cosmetics desire for the perfect figure. Now, I'm not going to talk anything about cosmetic aspects of obesity today, except to give you one tip. If you want to look thinner, hang around people fatter than you are. So, obesity is important as a public health a problem, largely because of its secondary consequences. And a major secondary consequence is type 2 diabetes, which 80% of people with type 2 diabetes are obese, and obesity is strongly associated with it. And type 2 diabetes is going to ravage the planet soon. There'll be at least 400 million people with the disease, mostly in resource-poor settings that don't have the chronic disease management to be able to deal with the amputations, the renal failure, all of these disorders that will really be a big challenge to these countries. And the increase in prevalence of diabetes over the last several decades, indeed even longer, has been accompanied by and probably driven by an increase in the prevalence of obesity. I mean, just one very quick piece of data. There's a very good data survey from 1890 in America where about 3.5% of American white males were obese then, and it's tenfold increased over 100 years. We've seen about a doubling over the last 20, but really taking even a 100-year perspective is very short. We've been evolving as primates for 85 million years, and it's only in the last 100 years that we've seen this enormous challenge to our species. And that clearly has got nothing whatsoever to do with genes. The change in obesity prevalence certainly over the last 20 or 25 years, has happened so quickly that it can have nothing to do with our genes. We don't transmit them that rapidly, and we don't certainly evolve them that rapidly. And it is clearly related to the availability, the advertising, the portion size, the palatability, and the ubiquity of highly attractive energy-dense food, combined with the lack of necessity to expend any calories in order to achieve that, but either in the workplace or in the domestic setting. So it doesn't need a genius to work out that we have created an obesogenic environment. And we can certainly talk afterwards about the nature of that and the precise things we might want to do about that in the future. And the best scientific data that environment matters is when you move from a low prevalence area to a high prevalence area. Japanese migrating from Japan to America, they tend to acquire the body mass index, the adiposity, closer to that of the society into which they move. So clearly your environment does have a big effect at a population level on whether you're obese or not. But, a sizable question is, if that's true, then why isn't everybody fat? Because we have a very obesogenic environment, and yet even in places like Louisiana, where 40% of people are obese, but at least 30% of people are lean. Are those thin Louisianans? There's only two real conclusions you can make. Either they're morally superior, or they're biologically different. Now, as someone who's always been a bit circumferentially challenged myself, I'd like to sort of think that there might be biological reasons why some people are susceptible and some people not susceptible to developing these disorders. And unfortunately, there's very good evidence that there are biological features which determine who is susceptible and who isn't. Now, a really good way of finding out how genetics impacts on any biological measurement, whether it's height, whether it's IQ, is to compare identical twin and non-identical twins grown up in the same family, or indeed to look at adoptees and see if they're more like the adopted family than they are their biological family. And there have been at least 50 studies of identical twins versus non-identical twins over the last 60, 70 years, and they are remarkably consistent. They show that the heredity of your body mass index, the amount of adiposity you have, is one of the most heritable things we have. It's not quite as heritable as height. In a clinic, we use your mid-parental height adjusted for sex as the predictor of what height you're going to be. We all accept that you tend to be somewhere in the middle of your two parents. Adiposity, i.e. the amount of fat you carry, is not quite as strong as that, but it's very strong indeed. The figure varies somewhere between 0.5 and 0.7. And so explaining a large proportion of the variability between individuals. Now, what I mean by heritability is if I took everyone in this audience and took the leanest to the fattest and then picked two of you out and said, what's the difference in the weight of your fat mass? What's that difference? And measured it and said, the difference is 30 kilograms. What percentage of that difference is down to genes? And that was what I mean by that number, that percentage, means the percentage of the difference between people. So that means that if this was an audience 100 years ago, you'd probably all be a lot leaner. But 100 years on, the whole distribution has been pushed up. But where you are on that distribution is largely or very strongly determined by your genes. And Jane Wardle, in fact, even more recently has studied 11,000 twins in the UK around the age of between 10 and 12. And the heritability is, even in the midst of this obesity epidemic, it's still very strong. There's been no real change in the estimates of heritability. And you can see here on the top here, these are pairs of sisters. They're born on the same day, they're twins, but they're non-identical twins. They carry only 50% of each other's genomes, and they come in all shapes and sizes, as you know, brothers and sisters do, because half their genes are different. You take these, and these are sisters born on the same day, but they share 100% of their genes. Hard to look at those pictures and say, I don't believe genes have got anything to do with body fat and how much. I mean, they are remarkably consistent, and there is a very strong concordance between identical twins for adiposity. And the most dramatic piece of data from that is a chap called Mickey Stunkard. He's in his 90s now and still active as a child psychiatrist in Philadelphia. And he realized that in Sweden in the 1950s, the sexual revolution preceded the contraceptive revolution, and lots of children were born out of wedlock. And many of them were or some of them at least, were twins, a reasonable proportion. And the Swedish authorities then thought it was a good idea to separate twins at birth and bring them up in different families and not tell them that they were twins. So he realized that later, as they became more enlightened, and they began to understand and broke the code, and they began to know that you could go back to the database and look at the heritability of adiposity. And indeed, when he looked at that, that he found that it was remarkable. There was no correlation between the adult body weight of the children and the families they were brought up in, but there was a very powerful correlation between the twin that they'd never seen. So I'm going to try and tell you that there are inherent biological processes strongly influencing the amount of fat stored in animals, including ourselves. These processes work with varying efficiency between people. Inherited factors play a major role in determining the variation in efficiency of those processes. And if we understand that biology, we might be able to help us to do something about it. So the cause of obesity is actually remarkably simple. we can all go home now because this could be the end of the talk. It's energy stored is energy in minus energy expended, and that really is pretty much it. But you can actually, of course, you could, when you have extra energy, you could store it as fat, or you could store it as something else. It'd be wonderful if you go out for a pig-out meal and put all that into a vigorous beard growth that you could cut off in the morning. But unfortunately, we're not really, we don't regulate that, and wouldn't be too popular with some elements and members of the audience. But fat is a very efficient way of storing nutrient, and of course that's why we evolved, to store it in conditions in terms of food availability, so that when we had periods of food deprivation, we had a source of very dense, packed energy to keep us going. But we kind of forget how remarkable it is, how sheer amounts of calories that we put in and out of the body during our lives, and how, despite this huge amount, we don't oscillate between Kate Moss and Luciano Pavarotti all our lives. Most people tend to stay along somewhat similar trajectories. If you tend to be a very lean person, you tend towards lean trajectories most of your life. If you tend to be obese, there is, of course, some wobble, but by and large, people tend to have a degree of stability in their adipose stores throughout life. And the first experimental work to really test that in animal systems was a chap called Kennedy, a British physiologist who studied rats. And essentially what he did was he overfed by gavage feeding, gave rats more food, made them fatter, and then just asked the question, if I stopped that after 2 weeks, would they just stay that way and keep growing throughout? By the way, rats keep growing all their lives, so if you see a really big rat, it's a really old rat. And what happened when he stopped was that they underate and got down to the trajectory that they were on before. And similarly, if you deprived rats of food and for a couple of weeks, just gave them half rations and then put them back in their original cage, for the next week or so, they overate to get exactly back to the trajectory that they were going to be on before, showing experimentally there was some sort of sensor system in an animal, a rodent, saying that it wanted to be at a particular level of weight and body fat stores. And we sort of knew that was likely to be in the brain, really from human naturally occurring experiments of nature in the 19th century, when perfectly healthy, fit adult patients suddenly started becoming grossly obese and grossly fat, and were found at post-mortem after they died to have massive tumors in the area of the brain called the hypothalamus. And those individuals was really the first instance that it was an area of the brain, that particular area, was an important area for controlling body weight. And the first half of this 20th century was an amazing time of experiments using mice, and then later genetically modified, naturally genetic variation in mice, to understand this central control of weight. But that culminated in the 1990s with the discovery of why this enormous mouse is more than twice the weight of its two otherwise identical twin siblings. So it only has one different gene change, one single nucleotide change that makes it twice the weight of its siblings. And that's because it lacked a substance, a protein substance, a hormone coming from fat tissue called leptin, which goes to the brain and keeps the brain informed that you have adequate fat stores. And if you don't have leptin, you turn into an aggressive eating machine. Essentially, the brain perceives itself as starving, and the animal continues to eat voraciously all its life and becomes hugely obese. And that completely transformed the view of obesity, turning it from a sort of vague science, to a really hardcore. There was at last a molecular handle on which we could understand a way into this system. And that led to a wonderful flowering of science over the subsequent 20 years, understanding both the long-term signals, whereby we understand fat stores and how they work in the brain, and the short-term signals that come after we ingest food to say, oh, I've had enough of that burger, because there are different signals that come from the gut. So there's long-term signals telling us how our long-term fat's stored, and then there's short-term signals saying that meal's big enough, that meal's got the right constituents. And we're beginning to understand a huge amount more about the nature of those signals. Was any of that relevant for humans? Well, that's, I guess, where I wandered in entirely by accident through this patient who doesn't mind me showing her photograph. Indeed, we've been mates for nearly the best part of over 20 years now. And that's Yvonne on her 50th birthday. But this is Yvonne when she was 2.5 years of age, when she was admitted to the Queen Elizabeth Hospital in Hackney for a year to stop her becoming fulminatingly obese. The parents were unable to control her weight and her appetite. So she was admitted to hospital for a year, and she remained obese all her life, and then presented to me later on with a lot of different endocrine abnormalities. And eventually we did discover that she had a defect in the ability to make small hormones from big hormones, and we discovered the nature of the gene defect a few years later, and she was really the first human in which a deep understanding of the biology of this gene really gave us an insight into why someone could become obese simply because of a molecular defect. And because of the recent discovery of leptin in mice, we went aggressively looking for evidence that this might be important in people, and found the first children, human leptin-deficient children, who really had the same phenotype, the same phenomena as we've seen in the OB mice. These children were very obese from a young age. They were constantly seeking food. Their lives were dominated by food-seeking behavior, and they were massively obese by the age of even 6 to 9 months. And we discovered that, my colleague Sadaf Faruqi, we discovered that these children were similar to those OB mice. They lacked this hormone leptin. And that initiated for us a very exciting phase of research where we'd gone from not really understanding at a fundamental level any human being's obesity to discovering a whole range of genetic defects which led to children becoming severely obese early in life. And we had no real reason to understand, to believe. We had no prior thoughts as to where those genes or where they would be acting. But we found a whole series of genetic disorders. And when we found them, what we discovered was really two things. One, those gene products were almost always working in this part of the brain, the hypothalamus. In pretty much all cases were. That's where their main functions were. And secondly, when we took the children into the clinical research facility and studied them in detail, they were obese largely not because they were burning off calories slowly, although some do slightly have a slow burn, but mostly the most dramatic abnormality is when we gave them access to completely ad lib food, and they ate vastly more than you would expect for their degree of lean mass. These children's appetites were stimulated and their satiety was impaired. They were unable to sense fullness, and they were unable to control appetite. So in effect, these obesities were not a metabolic disorder. These were a neurobehavioral disorder associated with hypothalamic dysfunction. And gratifyingly, in the case of leptin deficiency, it's a very rare condition. We're treating about 20 of these children worldwide. But in every case, when we give back leptin, we completely reverse all of the abnormalities in these children. And in some, it's enormously gratifying because they really have phenomenal clinical and behavioral responses. And essentially, within 2 days, they start eating less. Within 3 days, they're eating 70% less than they were eating before they started. And within months, they sort of totally normalize. And leptin has profound effects on the higher brain functions, the sorts of parts of the brain that are associated with addiction and reward, even before they lose weight. Within a few days of giving leptin, if we show pictures of food-related stimuli to these children, if we show them in the absence of treating leptin, their brains light up like a beacon. But within days of giving back leptin, that's totally normalized the brain's response. So here we have this real evidence of this lack of a Cartesian dualism, your brain and your periphery. Here we have a simple hormone circulating, coming from your boring fat cell, and yet it's having profound influences on how your higher parts of your brain are interpreting food-related stimuli. Have we stopped finding genes for obesity? No, we haven't. We continue to study over 5,000 of these children with severe obesity, and in work now led by my colleague who was my fellow and now a professor in her own right and leading this work, Sadaf Faruqi, and my colleague at the Sanger Institute, which is a major genomics institute, we are finding many, many more genetic disorders causing obesity. They range from transcription factors preventing the development of part of the hypothalamus to signaling molecules downstream of leptin, SH2B1 and KSR2. This genetic defect actually does cause a subtle decrease in energy expenditure. These people are more energy efficient as well as being more hyperphagic. And we've just completed sequencing of 1,000 exomes in many of these children, and some fascinating groups of disorders emerging. We find a very strong enrichment in proteins associated with neuronal development. The semaphorins, alterations in those proteins are very heavily enriched in this obese group, suggesting that the wiring of the hypothalamus may not be normal in many of these children who don't have some of the typical disorders that we described in the previous decade. So it's we're still in a rich seam of discovery here in terms of understanding. And I'm afraid there are many ways of getting obese, because we know if you simply injure a hypothalamus, or you take a hammer, have a car accident and you damage it, the default of a damaged hypothalamus is to eat more. So your brain, so the default, so anything that really causes severe damage to this area of the brain will release appetitive behaviors and make it very difficult to control obesity. Are these relevant to common disorders? I'm not going to talk much about the genetics of common, but it has engendered a really new major collaborative ethos with huge numbers of people being studied. And this is now asking whether these conditions these rare conditions have any relevance to the general run-of-the-mill weight variation in this room, for example. Do genetic variants in those molecules have any relevance here? And the actual answer is they do. Many of these genes in this bin here, where subtle variants cause subtle changes across populations, are the same genes where major variants cause major MC4, NEOJ1, SG2. So what's relevant to the biology of these extremes is in more subtle forms relevant to here. What's interesting about this group, many of these genes, in fact, the majority of the genes, when you look at just weight across a population or body mass index, most of the genes are indeed genes expressed in the brain. And they're not really inflammatory genes, and they're not peripheral genes. If you look, however, at waist-hip ratio, whether you're an apple shape or a pear shape, there's almost no brain genes in there. So it's almost as if your brain is setting the set for how much energy balance you want to be in, and your stuff below the neck is determining to a large extent where your central, whether you express central or peripheral adiposity. So I couldn't resist this little bit. There's a little bit of digression, because I know this was called the Fat Lab, and I have a vet in my lab called Eleanor Raffin, and a few months ago, I supported her to decide she wanted to work in, she thought it would be quite interesting to work in some veterinary models of obesity. They might give us some insights into humans and it might be quite useful. So here's a fat lab. And it's the most common obese breed of dogs, and 40% of mature Labradors are obese, and the owners, if they're really honest, they say they're greedy. Gosh, you just really have to keep them. You really have some nods in the audience coming from people who have greedy Labradors. And so dogs are wonderful for genetics because they come through fantastic bottlenecks. So you can do, with very small numbers, you can do genome-wide and find areas of linkage to disorders in trivial numbers of animals. So we decided we'd go ahead and find some fat labs and some lean labs and just have fun and see if we could find some genes. And we got fat labs and we got lean labs, and we only got about 20 of each, but then we had a young undergraduate student in the lab who wanted to learn how to do some DNA work, so gave her a few genes to screen, and lo and behold, she came back and said, excuse me, I think I found something interesting in this gene POMC, and we know that if you lack POMC in humans, you're very, very obese. It's downstream of leptin in the brain. And she said, in one of the dogs, you just found this whole deletion. This is a 14 base pair deletion. This isn't just a subtle, this is a complete chunk missing from the genome of this Labrador. So we said, we better go and check it in all the Labradors. And you checked it, and it's hugely enriched. You see the deletion, these are the obese dogs, and these are the lean dogs. So it's 4 to 5-fold enhanced with a very high statistical chance. So it is present in some of the lean dogs, but it's an intriguing, and it's a major deletion. 2 of the most obese dogs are homozygous for this deletion. And we know that humans who have point mutations in this gene do get obese. And what's nice is that it's not present in the mouse or the rat. So we don't have any animal model of this human disease, but we may now, in the dog, have a nice model in which we can and also very attractive therapeutic target, because there's lots of ways we can replace the missing peptides. So we may end up curing fat Labradors before we cure fat humans. So in the last part of my talk, I'm going to, this bit of my talk, I'm going to move from the causation of obesity to the consequences of obesity. And really, the link I mentioned at the beginning, the whole drive for this is that we're not so bothered by the cosmetic aspects of obesity, but diabetes is a really serious illness, and diabetes is strongly associated with obesity. So how does that work? And I think when I hear a lot of public debate about it, I think there's a lot of confusion and a lot of oversimplistic thinking about how this might work. And I think it's useful to break it into 4 sections. So the most important thing you can try and do is keep in energy balance. So energy balance is maintaining your energy expenditure the same as your energy intake, and therefore not gaining weight. And if you do that, and that's largely, or as I've tried to show you from our genes, it seems to be that the biology underlying that process tends to be very brain dominated. But if you can't do that, then you really want to safely store your excess calorie in your fat tissue, and you want to have safe rather than unsafe fat. And that's, I think you probably heard quite a lot of that at this exhibition. There's been a lot about fat and its biology. So you really want to store it safely in fat. Now, if you don't do that, then you start getting trouble in your muscle and your liver. And they are the two tissues that most respond to insulin and clear glucose. So you really want to try and keep these tissues as insulin sensitive as you possibly can, or else your pancreas has to work really hard and secrete loads of insulin every time you have a meal, and eventually your pancreas will get exhausted. And once you exhaust your pancreatic beta cells, then the glucose rises and then diabetes supervenes. So it's a complex process, not a unitary one, and you can probably break it down into many of the, and some people, probably a lot of people wandering around who get to one phase but not the next. And genetic factors are very important in determining who has susceptible beta cells and who does not have susceptible beta cells. So just to take that first step of going from energy balance to fat storage, if you asked 100 diabetes researchers now, what is it that makes you sick when you get obese and you get, we have to really understand how this sustained energy balance leads to this insulin resistance in liver and muscle. And most people say, oh, the bad guy is the big bad fat tissue, because now you've got these big inflamed fat tissue and it's producing lots of stuff, and it's those stuff that are going to liver and muscle, and it's the mediator, it's the culprit in leading you from obesity to its adverse consequences. But actually, there's an older idea, and one that I think has got less traction in some ways, worth really thinking about. And that is that your big fat cell is not actually your enemy. Your fat cell is your friend. If you're facing sustained positive energy balance, by far the safest place to put it is in your fat tissue. And it's only when you start reaching the limits of storage, of safe fat storage in your fat tissue, and you start redirecting these nutrients and ectopically depositing them in muscle and liver. The real culprit in all of this is this ectopic nutrient deposition in tissues in which we're not designed to store those fat, particularly those fat-related nutrients. And I'm particularly predisposed to thinking that way because we run the national clinical program for managing people with a disease called lipodystrophy. Now, these people may look thin to you, but they're not thin. They actually don't have functioning fat, or they have inadequate amounts of fat. They cannot make fat, or they cannot make fat within their fat tissue. And far from being healthy and muscular as they may look to you, they all die young, and they die young of accelerated metabolic syndrome. They all have severe diabetes, severe insulin resistance, severe triglyceride raising in the blood, severe cardiac disease, and very few reach it into healthy middle age. So it's a very dangerous condition, not having fat. And we, particularly my colleague David Savage and I, we've discovered many of the genes that cause humans to be lipodystrophic, and there's a whole range of things impacting fat synthesis and fat cell development. But I just wanted to tell you about this amazing thing. This is the fat cell droplet. Now, fat cell was once thought by most cell biologists to be the most boring cell in the world, because you look at it and it's 99% one drop of oil, of triacylglycerol, described as a fried egg on a beach bowl. So it's an enormous cell, and the nucleus is the fried egg, and a thin rim of cytoplasm, and this enormous, boring, homogeneous droplet of fat. And that's what a fat cell looks like. But actually, this is one of the most fascinating organelles in the body. It's beautifully controlled by a series of proteins that attach to its lipid monolayer. And that set of proteins is highly regulated and is designed to release free fatty acids into the body when you're starving, and to stop releasing them when you've eaten. And it's a very simple but beautifully coordinated set, which have these enzymes which do the job, and then these other proteins, I think proteins called side C and perilipin, which, if you like, are the conductors of the orchestra. They're the ones that organize these enzymes to do their function or not do their function, depending on whether you're starving or you're fed. Very simple system, but beautiful. Now, until recently, we didn't really know what happened when that went wrong in humans. But David and I have found some human beings who lack these proteins, or lack the functional variants of those proteins. And these proteins don't work anywhere else. They don't do anything else in the body. All they do is control the breakdown and non-breakdown of this fat droplet in white fat cells. And what are these people like? Well, these people are all severely insulin, exactly the lipodystrophic syndrome that I described to you. They all have diabetes, vascular disease, hypertension, fatty liver, very sick from a young age. And in both of these cases, the only thing that's wrong is their inability to safely store fat in the white droplet. And David has just recently made a mouse with this mutation. And in that mouse, he can cross it with this OB, the enormously fat OB mouse. And it makes it thinner because it can't make fat cells properly. And it makes the fat tissue look really healthy. The fat tissue is not inflamed, and there's no inflammation. In fact, it reduces the inflammation. And yet the mouse has got severe diabetes and metabolic, much worsened in the absence of any inflammatory signal at all. So really all you need to cause all of the metabolic syndrome is that your fat cell, which is the topic of this wonderful symposium here, your fat cell not working properly and not doing its simple job of storing when you're fed and releasing when you're starving, not doing that is enough to cause everything that we fear, all these diseases that we worry about. So I'm going to end with a more general sort of talk and think, what are we going to do? Does this biology have any relevance to disorders? Well, is it like smoking? Can we have the same solutions as smoking? Can we have successful influence on individual population behavior through information, facilitation, incentivization, and coercion, the various aspects of public health people try and do at various levels to try and make you stop doing things that you shouldn't be doing? And of course, it's tricky because with food is different. We have to eat. We don't have to inhale the rolled up leaves of nicotiniana, whereas we have to actually eat. So maybe I think hypertension might be a nicer analogy. And I'll just remind you, people don't think about blood pressure much anymore, and that's largely because it's a partially or at least mostly solved problem. Back in the 1950s in the British Health Service, the wards were full of patients with severe uncontrolled hypertension. Really many strokes, many heart attacks, many renal failure. It was a huge medical problem. A large percentage of beds were occupied with people with, even when I started in medicine, it was still a sizable problem, malignant hypertension. It's now effectively gone. Now, how did we do that with high blood pressure? How did that get solved? Well, hypertension's got some nice analogies to obesity, because hypertension is just being on the higher end of a blood pressure distribution in the general population, and obesity is just being the higher end of an adiposity distribution. So they're kind of similar in that way. In the 1940s, the only thing you could do when you were severely hypertensive was go on an appalling rice diet. Almost no one could tolerate it. It was dreadful. It was terrible. And then in the 1940s, when people were dying of severe hypertension, they did some drastic surgery. Cut your sympathetic nerves, and it was great, your blood pressure dropped, but you could never stand up again. So then you had severe side effects, terrible drugs, gave you terrible depression, terrible postural hypotension. Then we started having slightly better drugs in the 1960s, and then we started understanding the biology of how blood pressure was controlled and how the kidney transporters were important, and then from the 1970s, based on a real understanding of the control of blood pressure, we gradually developed a whole series of agents associated with a fundamental understanding and a gentle tweaking of the system. And now, in effect, there is nobody whose blood pressure can't be controlled by drugs that cost pennies. And that has been combined with the public health message of reduction of salt and a public health impetus to reduce dietary salt. And that combined public health and pharmacological approach is really what's led to hypertension being a major and being solved. How about obesity? Where are we? Similarly, I think we're just many decades behind what's happened in hypertension. We know that hypocaloric diets and exercise can work, but I find it very hard to be sustained. Here's a superb psychiatrist, researcher, Chris Fairburn, who developed wonderful treatments for anorexia nervosa and for bulimia, but he took on obesity recently, assuming that kind of would be easier than anorexia or bulimia. And in conclusion of his trial, he basically said that obesity is resistant to psychological methods of treatment, and wondered whether it was ethically questionable to claim that psychological treatments for obesity work. So, not a great promise for obesity, not for the other eating disorders, but obesity may be different from these. We're at a phase of bariatric surgery, which may be our current similarity to the surgical sympathectomies of the 1940s. Wonderfully effective, but can have some severe side effects. We have drugs with limited side effects and have to be dropped, and significant adverse effects. So our major impact on hypertension has been policies to reduce salt intake, screening, and now drugs that are generic, cheap, and in combination. And I'm hoping that if we're optimistic, we can look at obesity taking a multidisciplinary approach, public health measures, the selective use of cheap and safe medications based on an understanding of the biology of the system. And it's early days yet. We really discovered that first home only in the late 1990s. This is very recent science. So I'm going to end by saying that the unique thing about obesity is that despite being a major and life-threatening disease, it is one of the few human phenotypes that people still feel completely comfortable about vilifying the sufferers. And easy to pick up newspapers and find journalists casually talking. Although it always amuses me that journalists are often never the slimmest of individuals themselves. But it's sometimes quite horrific. If you just read this, and it is quite amusing, but then you start thinking back, can you think of any other type of human that this sort of language would be tolerated? If this was written about any other group of human beings, would we tolerate that? This is a medical doctor writing in a medical magazine. Quite extraordinary, really, that we tolerate this sort of vilification of a group of human beings. So this is Yvonne, I told you about before. And on her 50th, she wasn't too well. She was on 200 units of insulin a day, and she nagged me about bariatric surgery. And I said, Yvonne, it's not going to work because you've got this genetic condition causing your obesity. So it's definitely not, it's not going to be effective. But finally, I gave in, and she underwent bariatric surgery. And here she is in Hong Kong on a cruise, having lost 40 kilograms, come off insulin, no longer diabetic, and remains so 5 years later. Hugely successful, despite the underlying problem of her genomic variant being the actual cause of her obesity. But still, the signals that she gets with the rewiring of her guts are sufficient to allow her to maintain normal health and normal bariatric. So, on behalf of Yvonne and other great fatties of the world, including Thomas Aquinas and Oprah and Helmut Kohl, we'd like to say to Dr. Bob Berry and his ilk. So I'd like to just thank the various people who have funded my work, and my colleagues, of course, and patients and families. But recently, I've had to add another. So thanks for your attention. Thank you.
So, wow, in terms of Fat Lab and the science of fat, and it's official. It is good, and it's even beautiful. The fried egg on a beach ball concept is one that will stick with us. I mean, I know from just looking around, there are people who are going to want to ask questions, and we will open this to the audience in a bit. But I do want to take the opportunity to ask you a few questions first about your own journey from Dublin to knighthood. I mean, and I know it's an embarrassment. I'd much rather just get on with the science and the clinics and so on. But it is fantastic for Ireland that you've done what you've done. It gave everyone a big lift when it was, announced. And so, you're Finglas originally.
Yeah, I suppose, north side all my life. When I was in Artane for the first couple of years of my life, then my dad got a chemist shop in Finglas, and we lived above the shop until I was about 11 or 12, and then moved up to Ballymun, which is now Glasnevin North, of course. It was Ballymun Avenue when I was there.
And your decision to do medicine, when did you?
That was always wanted to do it. Liked the idea, liked science, liked the idea of the humanitarian aspects of medicine. So lovely combination. Never regretted it, and yeah.
And I met a classmate of yours a few months ago, and he said that after pre-med, the first year, 6 you went on holidays down to the west of Ireland, and you took your
such
first
a nerd.
med,
I'm not honestly.
You took your first med books and were reading them on the beach. Is that true, or is that legend?
I really wanted to understand physiology. I really liked physiology.
Okay.
I mean, I was dying to understand how the human organism works. I thought, well, you can read other books and read some human physiology, because it's very fascinating. I suppose. So, sorry, I was a nerd.
No, it's okay. It's okay. And in fairness, he is working in Castlebar, and you are working in Cambridge, and so from medicine, then, why I mean, I'm an endocrinologist. I love hormones. I went into it because growth hormone actually was just coming. Recombinant growth hormone was just available when I went in, so I got into a project and I loved it.
So there's negative and positive reasons for endocrinology. I've got 10 thumbs, so any trade use would have been useless in anything with my hands. So I had to do something that was just involving.
Your shoelaces are
sorry, yes, they are.
And
so I'm totally
it's an endocrine
useless.
thing.
Yeah, it's an endocrine thing. So I knew I couldn't do anything that involved manual dexterity, and I just applied biochemistry. I loved the idea of stuff circulating around. Amazing. I remember reading as a kid, an article on hormones and how they produced in one place and communicated with another and integrated the functions of the whole organism. I found it just thrilling. So I've always enjoyed endocrinology, and that diabetes. I was going to be an endocrinologist, but then a man called Robert Turner convinced me that insulin was the most interesting hormone, and that I should think of diabetes as a hormonal disorder. And I guess what happened with obesity is, as soon as it became, I mean, I'm probably pretty blinkered really. I only got interested in it when it had a. I love this idea of small bullets flying around the body, telling one part of the body, using quantitative information to tell another part of the body how to behave. It just seems to be an extraordinarily beautiful, sending these bullets around through the blood system and flow. It's an information flow. That's really what it is. It's a circulating information.
I mean, I have kind of read one or two interviews you've done where you've talked quite a bit about beauty in science and likened it to music and that. I mean, medicine and science versus art. Where are you on that? Do you think we're too rigorous around statistics and not?
No. I mean, I think anyway. The lovely thing about medicine is and doing medical science is that it can satisfy a lot of the parts of you. I mean, if you doing a decent clinic and helping people is a lovely thing to be able to do. But if you're doing research too, and you can see, I mean, the great thing about science as a human activity is it allows you to see things that no one's ever seen before. It's the modern day exploration. And I guess that's what artists through exploring their art or their music, and what scientists and explorers used to do. It's harder to explore the planet now because most places have been explored. But through science, you can open up something and go, wow, no one's ever seen that before. And that's a fantastic thrill. It's a different thrill from medicine, and so I think one of the advantages we have as doctors is that we have access to human variation, and we are effectively human. We have unique ability to be human biologists because simply varied human biology presents itself to us because people are ill and they come to us, and yes, we can deal with them compassionately and with affection and with empathy, but it doesn't mean that we can't be curious about understanding at a deep level, an atomic level even if you like, what the nature of their dysfunction actually is.
And overall, I mean, you have gone from human observation back into, if you like, the lab-based animal knockout. My own view is we need much more of the human back into the lab to test it in the sometimes controversial animal setting. I have had difficulty getting things published because they're not relevant to the mouse.
I wouldn't like to set up this false dichotomy between people who work on mice, people who work in humans. We discovered a lot of this stuff that was discovered about the control of body weight was discovered in the mouse, and almost all these genes that we found that affect hypothalamic function, if you make a mouse with the same gene, you get an identical or very, very similar phenotype. Now that's not the same when you do it with insulin resistance. It's not the same with beta cell function. It's certainly not the same in the immune. So you can imagine the differential evolution of the immune system, very powerfully different between species. So there are some phenotypes in which interspecies difference is very powerful. There are some phenotypes which you can see in Drosophila or the zebrafish, or even in yeast. There are some phenotypes that you can share between humans and yeast if you disrupt certain things. So it all depends. The answer is, in science, the devil is in the detail. Yeah. And the idea that mouse is all bad and humans is the only thing to do is nonsense, because they're bidirectional. Okay, the work has to be bidirectional.
Just to wind forward a little bit then, you've gone down the endocrine route, you're a diabetologist, you're originally from Dublin, job comes up in Dublin, and you are applying, and you're considered a strong candidate, will I say. Your decision and the nature of that decision to not go to the interview, just tell us a little bit about that.
It was quite. I mean, I'd spent time in a successful time in Oxford. I'd then gone on to Boston, where I was learning that my 10 thumbs were not just an obstruction to surgery; they were an obstruction to doing high quality, challenging lab-based research too. And I wasn't doing terribly well. And I think, oh God, what am I good at? I'm pretty good at seeing patients, and I'm fond of Dublin. Why don't I just give all this up and just because I'm really struggling, and I only had a few months of my own funding left, and so I around the same time I asked my then girlfriend whether she'd. So I had a 48-hour period of asking Susie to marry me, sitting outside an interview in Dublin, deciding not to go, going back to Boston, changing labs, and sometimes in life, all your decisions come. So I'm in retrospect, I'm glad I did because I've had a wonderful marriage and a wonderful last 20 years in.
And the knighthood and the way you heard about it then, I mean, clearly, we've seen the work that's gone on, and that's as part of a team that you kind of represent, and we kind of know that, but you have been, how did you hear that you were even being considered for it?
You don't hear at all. Nobody, you're not supposed to know, and nobody, I had no idea. I was at a meeting. I think I misremembered that I was actually in a meeting in Canada, and my PA texted me or emailed me and said there's a letter from the Prime Minister's from the Cabinet Office saying to be opened by addressee only, and so they opened it, and she says, we'll open it and fax it or scan it, and of course the first question it asks is, are you British? To which the answer was no. So I had to then decide whether to go the Terry Wogan route or the Bono or the Bob Geldof route.
And meeting the Queen in person?
Great fun, very impressive. I mean, I know she's been here and did wonders for Anglo-Irish. She is a phenomenon. She's 88 years of age. She stood up for an hour and a quarter, talked to 90 people, managed to, I mean, I don't think I could manage that. It's a pretty amazing feat for someone of 88 years of age.
When she visited Ireland and Trinity a couple of years ago, she bounced up the steps like she was, in her 50s. If you were to look back at a kind of a tipping point, you mentioned that 48 hours kind of where a whole lot of decisions and a change of lab. At what point did you feel that your research and your capacity to lead it and generate it was now up and running and completely?
You never do. You always feel as good as your last paper. So I'm not. You don't get if you get complacent like that, you're dead. So no, I've never got. I still feel It's a bad case of the imposter syndrome, which is a very healthy thing to have.
I agree with that. I had the opportunity to meet Dr. Margaret Chan, the Director General of the World Health Organization, yesterday, because they've started a childhood obesity campaign, commission to end child obesity, a bit like their Health for All by the year 2000. And they're focusing, as they have to, on the environment and the environmental measures that you mentioned. I mean, I'm kind of on one at the moment about sugar-sweetened drinks and Coca-Cola sponsoring the Dublin City Bikes as of today, putting their brand out there. Where are you on that kind of issue?
So I always try to be with the data rather than with any presupposed, I think the problem of nutrition has been bogged down and disempowered really by over-passionate advocates on one side or the other. And I think you just need to be and it's very hard to measure what people eat in the free world. So actually getting data is really hard. So there are very few randomized controls. It's very easy to do a randomized controlled trial of a drug, very difficult to do a randomized controlled trial of a nutritional change. But where I stand on sugar and non-sugared beverages is there's two pretty decent RCTs in children where their beverages were randomized to nutritive and non-nutritive sweeteners. It looked pretty convincing to me that after a 6-month period, if you were randomized to the non-sugar sweetener, you were about a kilogram lighter after a year. If you extrapolated that across the population, that would be a huge health benefit. So very broadly, if you want me to oversimplify, I think a simple one, it wouldn't be a solution, but a simple thing to do, which would be cost neutral for Coca-Cola, would be to tell them that we could put a 50p tax on sugared beverages and give a 50p supplement, give them a 50p incentive to make non-nutritive sweeteners. Because I don't see any real data that non-nutritive sweeteners have any serious adverse health consequences.
In fairness, it is Coca-Cola Zero that they've gone with on it, which is their, the WHO view is that is brand positioning. So McDonald's are the biggest marketers of fruit and veg in the UK because it allows their brand at kids' TV time.
Well, there's a wonderful thing that Teresa Marteau talks about caloric discounting. So if you go to a McDonald's and you get a salad, you think that's healthy, so it means you can upgrade your fries to a large fries because the salad has the concept of negative calories. So fruit and vegetables. so there are all sorts of stuff that goes. So I think there's no incompatibility with what I'm talking about variable genetic susceptibility and biological processes controlling. No conflict at all with the notion that the environment that we've created, where instead of having 8-ounce Coke cans as when it was first started, we can go to a cinema and have 48-ounce Coke buckets. For a similar percentage of our weekly income, is a mad situation to have got into, and that these things are ubiquitous. There's constantly marketed. They're everywhere. It's hard to avoid. In the hospital, I have practically kilogram bars of chocolate offered to me for pennies. When I go and try and buy my sandwich at lunchtime in the hospital, I think how mad is that?
I mean, just today, New York, the High Court in New York overturned the ban they had on 16-ounce. So they are now in New York allowed to go back to the 48-ounce servings.
Oh, they did. So they
yeah. So they are now rolled. So New York Department of Health wanted to ban the big ones. No, it had been banned, but now it's been overturned. And that's
very powerful lobby. It's a very powerful.
Stunning
lobby.
Because it's an absolute no-brainer. But anyway, we have to stop. I think fat has arrived, and the fact that there are 100 people on a Friday evening in the Science Gallery as we kind of come to the end of the Fat Lab is a real marker of that. It's been a privilege for me to be here this evening. I would like to thank everyone for coming. That's number one. And number two, Professor Sir Steve O'Rahilly for giving his time yet again to Dublin and supporting Dublin yet again as he does consistently, and I hope will continue to do. Thank you very much. And good night. Thank you.